All right, so Helen is here. Hi, Helen.
Hi, Raj. How are
How
you?
are you? Good, good. And we're waiting for Yalda, I think.
She's last, so.
Yeah. Is Steven still, I assume it went virtual. Is it still virtual predominantly, or what's the?
Predominantly freshmen, and now just this semester seniors have been allowed to come back, but it's still hybrid teaching mode,
Okay.
both.
And what's
I'm
the
hoping
plan
by
for the
the
fall?
fall we'll be back
Yeah.
fully.
I just want to take a quick look through the attendee list to make sure we don't have anyone that needs to be on this side. And let's give it a minute before we start.
Okay.
All right, let's get started. Welcome to the faculty seminar session. This session focuses on neural engineering. We have 4 exciting talks lined up for you, and let's get started. The first talk is Professor George McConnell from Stevens Institute of Technology. George completed his PhD in bioengineering from Georgia Tech and then did his postdoctoral training at Duke. Dr. McConnell's research interests are in neural interfaces and deep brain stimulation facets of neural engineering. He's currently assistant professor of biomedical engineering at Stevens, as I said, and the title of his talk, if it hasn't changed, is, well, I can read it, Advances in Intraoperative Mapping of Deep Brain Stimulation Targets, Discerning the Sweet Spots. George, please.
Thank you very much for the kind introduction and opportunity to speak to you all this morning. As was mentioned, I'm going to be talking today about a very interesting application of neural engineering called deep brain stimulation, and this challenge of placing the electrode within these so-called sweet spots. I do also want to point out at the outset, for those of you who are unfamiliar with Stevens Institute of Technology, we're situated right across from Midtown Manhattan, and we do have several other brain-related researchers, including a recently formed Center for Neuromechanics. Use this to also pitch the announcement that we are hiring, and we're looking for a new chair of our biomedical engineering department, as well as a tenure track faculty in neural engineering. So please contact me if you're interested in learning more. I've broken down my talk this morning to tell you some background about Parkinson's disease and deep brain stimulation, or DBS, in case you're unfamiliar with that field, and some of the questions that I've been interested in answering and working towards answering in my lab. Parkinson's disease is climbing in the US as life expectancy continues to increase, with expected numbers over 1 million by 2030. It's characterized by what are called the cardinal symptoms of Parkinson's, including tremor, slowness of movement, stiffening of the limbs and joints, as well as these balance and gait symptoms that I'm especially interested in developing better treatments for. Currently, the best pharmacological-based treatment for Parkinson's disease is called levodopa or L-DOPA, It works very well early on, but in the advanced stages of Parkinson's, it can have its own debilitating side effects called dyskinesias. And you may have seen these if you've ever seen an interview with Michael J. Fox and him, those writhing dance-like movements in the chair are very painful, called dyskinesias, as I mentioned before, and can be more debilitating than the symptoms of PD themselves that I previously mentioned. Levodopa also doesn't treat all the symptoms of Parkinson's, including the balance and gait symptoms I'll be telling you more about. The technology for DBS is based on much of the same platform as cardiac pacemaker technology. It's developed by Medtronic, though other companies are interested in developing new technologies for DBS as well. So they're not the only player here. But the technology uses an electrode that's placed deep within the brain, as the name implies, and subcutaneously a lead is connected to an implanted stimulator that contains the battery pack and all the electrical circuitry used to deliver the tiny electrical pulses that are patterned to the brain tissue. And the idea, much like a cardiac pacemaker, is that the pathological rhythms in the brain are overridden by these electrical pulses, just like in a cardiac pacemaker where arrhythmias in the heart are paced over by the electrical patterns delivered. This is an example of the Medtronic's design of 4 electrodes, and a clinician programmer can wirelessly program the settings on these DBS electrodes to control the stimulus waveform, including the amplitude of the pulses delivered, the duration of these pulses, as well as the timing between pulses. Parkinson's disease is not well understood in terms of its etiology in the vast majority of cases. However, we do know that a small cluster of neurons here within this area, the substantia nigra pars compacta, degenerate slowly over time. And this loss of input to neuronal circuitry called the basal ganglia results in the symptoms you see of Parkinson's disease, where there's this area of the basal ganglia is known to be important for initiating and control of movement, and this control is lost in the case of Parkinson's disease. We've been interested in my lab in studying more about how deep brain stimulation works, and to do this we use an animal model, a rat model of Parkinson's disease, where we inject a toxin in one half of the brain. So you're looking here at a section through the face of the animal, a coronal section, and we've lesioned one hemisphere of the brain, and we can use histology then to look how many of those dopaminergic neurons were lost from the toxin and where our electrodes were placed. And this model allows us to not only test various stimulation patterns and play with these parameters, but also to study the effects on behavior and the effects on neural activity, and use that to understand how deep brain stimulation works. So what do some of the behavior actually look like when we apply DBS? This is an example of one of the behavioral assays that we use, where we monitor the circling behavior in these animals. And we've injected a toxin on the left hemisphere of the brain in this case. And without DBS, it circles to the left. And when we turn on DBS, you can see that the circling direction has now changed. So we performed this test across a large group of animals, and what we found was that higher frequencies frequency stimulation of DBS was more effective than lower frequencies, shown here, where 0% represents no change in circling bias. And this curve is similar to what has been shown in humans, where we know that higher frequencies are more effective at treating the symptoms of Parkinson's disease compared to lower frequency stimulation. This study was also my first exposure to the fact that the placement of the electrodes within the brain is a critical parameter. This is another study that we published showing that electrode location within the brain matters in this model. And these circles here are indicating correct placement in an area called the subthalamic nucleus, or SDN, which is the major location site for treatment of PD in humans as well. And we found that only when the electrode was properly placed here in the SDN did we get this effect of reduced circling rate with higher frequency DBS. So we've been using this model to design more reliable treatments for the gait dysfunction symptom of Parkinson's disease. And in the advanced stages of PD, this becomes a great problem because it can result in falls. So I'm showing here a human brain atlas and the advantage of another target here in addition to the STN, the SNR, that has been an area of interest for us. The advantage is that potentially in humans, you could apply DBS not only at the STN to treat tremor, but at the SNR, which has connections with locomotor regions in the brainstem and may improve these gait symptoms. And a previous study looked at this in patients with Parkinson's disease and quantified score associated with gait improvement. Initially, they found that SNR DBS had no effect on treating the gait symptoms. However, a closer look showed that the electrode was placed within the lateral part of the SNR. So part of my work prior to coming to Stevens was in using this animal model to look not only at the lateral SNR to improve gait in the Parkinson's rats, but also at the medial part. And this inspired an additional clinical study kind of supporting this work in animals, showing that medial sites within the SNR are more important than, maybe more important at treating gait compared to lateral sites. So this study again emphasizes not only, as I told you, the electrode has to be placed properly within the subthalamic nucleus, STN, compared to outside of it, but in this case, we saw that in another structure in the SNR, it matters not only being in the SNR itself, but being within the proper spot within the SNR, so-called sweet spot for treatment. So this brings me to the final portion where I want to tell you more about work to answer this question of reliably placing these electrodes. This is something that's not only of interest, of course, in my work with understanding DBS mechanisms in an animal model, but it's also important in work in humans with DBS implants. And current state-of-the-art for DBS surgeries is implanting the electrode using a stereotactic frame. They do preoperative MRI in addition to recording neural activity, so physiology imaging hybrid approach. And you can see an image down here of high resolution MRI being used to segregate where the electrode is within the brain and properly place these electrodes, in addition to this scribble nearby, which is the notes of the neurosurgeon as they lowered a microelectrode and measured the electrophysiology. So two different approaches. It remains to be seen whether imaging can replace microelectrode recordings. For now, microelectrode recordings remain used by a majority of neurosurgeons doing these DBS implants. So these microelectrode recordings are recording the spiking activity or action potentials from microelectrodes implanted in the brain. So we wondered if we could take this qualitative approach used by neurosurgeons and make that more quantitative to differentiate these structures in the SNR and know whether we're in the medial part of the SNR, which is more effective at treating gait, compared to the lateral part of the SNR to further look at mechanisms of DBS to treat gait. So we wanted to take this spiking behavior and quantify different features of that, correlate it with histology within the brain. This is another coronal slice, and we can fluorescently label our electrodes so that we know exactly where the tip was placed within these animals. And then we can map here the tracks as we entered different regions surrounding the SNR and within the SNR. And the red dots here are showing which sites we actually recorded spikes at. And you can appreciate here that a small percentage of our total electrode recordings actually even contained these spikes that we could then further analyze. However, the ones that did show spiking behavior, we found amplitude was a key feature in differentiating the SNR from the nearby structure of the ventral tegmental area, which was not where we were trying to place the DBS electrode, as well as differentiating the medial SNR from the lateral SNR. So this was some initial work, and you can read more about it along with some other freely available studies at frontiers.org. We next went on to look at LFPs, which we could potentially capture from every site. And we've been using a technique that we've developed called Spacer to look at these LFPs from sequential sites as we lower the microelectrode in the brain, capture the power spectrum at these different depths, and then use this analysis technique called coherence to quantify the similarity between the power spectrum at those depths. We can then construct these coherograms. So now I'm showing high coherence in warmer colors, lower coherence and cooler colors as a function of depth within the brain. And what we've seen is that these patterns look very different in the medial versus the lateral SNR. So we are very interested in pursuing this technique further. And our current interest has been also in collaborating with neurosurgeons who are interested in seeing whether this kind of approach could be used to differentiate other structures in humans.
George, just not.
So I think I'll stop there and thank everyone. So thank you very much for your time.
Thank you. Sorry about that. Excellent. Does anybody have questions? looking to see if anybody's posting anything. And
It looks like Helen raised her hand.
oh, wonderful, Helen. Go ahead, please.
Hi, George. That was a really nice talk, a very interesting work. I had a question about whether the SNR is very close to the VTA, the medial part, and how much do you think the effect might be related to its proximity to the dopamine cell bodies that might be somewhat spared in the MPTP, or I'm sorry, the toxin PD model?
Yeah, so what I showed you earlier with the atlas from the human, the STN is right next to the SNR. However, in the rat, that's not the case. So we think actually in the rat that we're not activating the STN, although that could be you're right that could be a potential.
Oh, I meant the VTA, the dopamine cells.
Oh, I'm sorry, VTA, yeah.
Yeah, because it's very close, the medial part, which is more effective that you showed, right?
Yeah, you're right. No, that's a great point, and we can't rule that out right now. So that's one of the reasons why we want to get more electrodes in the medial site so we can answer some of those questions.
Okay.
Good point.
Thank you, George.
Thank you.
Wonderful. Any other quick questions from anybody? All right. If not, shall we move on to the next talk? Our next speaker is Dr. Helen Schwartz. She completed her PhD in biomedical engineering from Arizona State University and completed a postdoctoral training at MIT. Dr. Schwartz's research interests are in multiple facets of neural engineering. One particular area that I'm assuming we'll hear about today is implantable devices with multiple modes of signal measurements. She recently joined our department of bioengineering at the University of Pittsburgh as an assistant professor, and the title of her talk is Chronic Recordings of Chemical and Electrical Neural Activity in Rodents and Primates. Helen, all yours. You are muted, yes.
Yeah, thank you, Raj, for the kind introduction. So today I'd like to present on our work, as Raj mentioned, to record chemical and electrical neural activity chronically in rodents and primates. My name is Helen, and I'm a new faculty at the University of Pittsburgh. So first, I'll briefly describe some of the motivation for this work, and then the methods that we developed to record dopamine from multiple sites in rodents and primates. We then developed microinvasive probes to enable these dopamine recording functions over longer chronic timescales. And then more recently, we've been working on multimodal interfaces to combine the dopamine recording with measurements of electrical neural activity. In particular, we were interested in the electrical beta band oscillations and the dopamine molecules, both of which we know are critically impaired in Parkinson's disease. And then finally, I hope to get to share some of the results we had with applying these systems in task-performing monkeys to study the role of dopamine and beta band oscillations in mood and motor control behaviors. So the motivation for this work is really that we know that a lot of the neurological diseases, psychiatric disorders stem from neurochemical dysregulation. And dopamine is a major neurotransmitter, neuromodulator in the brain. It's mostly found in this area called the striatum, here colored in purple. And in the primate, this region is composed of two subdivisions, the caudate nucleus and the putamen. And the dopamine that's released into this region comes from these ascending axons or fibers that originate in the midbrain SNC VTA dopamine neurons. And in Parkinson's disease, these axons die away, they degenerate, and this causes a massive loss of striatal dopamine, which leads to many debilitating impairments, movement impairments, as well as mood impairments. And these dopamine molecules are also implicated in many other disorders, and I've listed a subset of them here on the right. So in order to better understand the function of dopamine, both in healthy as well as in these disease states, we need to be able to measure dopamine in the brain. And the way that we do that is typically we implant sensors that are called carbon fiber electrodes into the brain. And these sensors comprise a carbon fiber, very small, 7 microns in diameter, that's threaded through a fused silica tube. And the carbon fiber acts as an electrochemical interface to detect electroactive molecules in the brain. The issue with these sensors is that when you implant them in the brain, you create a hole in the brain, as well as an inflammatory response, which you can see here as this and this amplified color changes around this hole, which represent increase in the expression of astrocytes, microglia, and blood-brain barrier permeability, which are all markers of inflammation. So what we want to do actually is to try to remove the scarring interface so that we can enable measurements, better, more accurate measurements over longer time scales. And the way that we measure dopamine is we use an electrochemical technique called fast scan cyclic voltammetry, or FSCV, where we apply this ramping triangular voltage every 100 milliseconds to the implanted carbon fiber electrode. And what happens is that when the positive going voltages are applied, the nearby dopamine will oxidize to dopaminoquinone, and during that process, electrons are transferred to the carbon fiber, and we can read this out as electrical current changes. Typically, the way that we visualize these voltage-dependent current changes is in the form of a color plot, where the color represents the current, the y-axis is the applied voltage, and the x-axis is time. And here in this color plot, you can see clear changes at 0.6 and negative 0.2 volts, which are the dopamine redox potentials. We can then extract these voltage-dependent current changes to look at the actual change in dopamine concentration. And this is the curve showing the extracted dopamine concentration change. And what's nice about this technique is that it has that fast sampling rate, so the 10 hertz sampling rate, to pick up the rapid dynamics of dopamine release and uptake, which occur on the millisecond time scale. And also, this technique affords a degree of chemical selectivity because the molecules will oxidize and reduce at different voltages. So in order to take measurements in vivo in animals, typically the way that this has been done is that we have to actually fabricate the sensors by hand. And so we create these fused silica threaded carbon fiber electrodes, and this can be a manually intensive process to build these small probes. And also, when each of these probes are implanted into the brain, into the striatum, the probability of being in a site of active dopamine release is only about 50%. So there's a very low yield in terms of being able to take measurements of dopamine. So what we wanted to do was to increase our success rate in picking up dopamine signals. So we made these microarray systems that would allow us to introduce multiple sensors into the brain. And also this would allow us to explore potential spatial heterogeneity of the dopamine release in the brain. And so here we took 2 of these microarrays that we fabricated and implanted them into the rat brain to record 16 channels, or from 16 sites at the same time. And the bottom left here shows the cyclic voltammogram. So here you can see current peaks at the 0.6 and negative 0.2 voltages, which are associated with the dopamine redox. And so this tells us that we're measuring dopamine. And then on the bottom right, these are the dopamine concentration changes that we measured as evoked by direct stimulation of the dopamine fibers. So at around 2 seconds here, we stimulated the dopamine neurons, and you can see that dopamine release occurs at about half of the sites in the striatum. And what was very interesting was that the dynamics of the dopamine signals are very different from site to site. And so we also wanted to do these multi-channel measurements in the rhesus monkey and the non-human primates. So here what we did was, because the brain is a lot bigger, we adopted a chamber platform that was originally developed in the Graybill lab where I worked at MIT. And we adopted this to introduce the carbon fiber electrodes onto the system, where we could then mount them onto microdrives that would allow us to move the electrodes to different sites over the course of time. And then also we could introduce acute recording sensors into the stratum as well to compare measurements from the acute sensors to the chronic measurements. And we can also introduce stimulating electrodes to directly stimulate the dopamine fibers. And with these systems, we were able to make the first chronic measurements of dopamine in primates, and we validated this with stimulation, with pharmacology. So we gave dopamine selective drugs that would enhance dopamine signaling in the stratum, and then also with behavior. So here we had the animals perform a task where we give them different sizes or different volumes of a liquid food reward. And we know that dopamine is involved in encoding some form of reward value and expectation. So we expected to see differences in the dopamine fluctuations based on these different reward volumes that we were giving to the animal, and which we did see. And so, so far I showed you that we were able to make measurements of dopamine over these chronic time scales. And however, one of the major challenges that remained was that the signals that we recorded at these chronic time scales, the amplitude of the measurements, as well as the frequency, the amount of dopamine transients that we were able to detect decreased over time. And so we suspected that a major factor in this overtime degradation was related to that scarring process that I showed you earlier, that inflammatory response. So in order to try to address this issue, we developed smaller microinvasive probes shown here on the left. So these are 10 microns in diameter. It's about the same size scale as the individual neuron cell body. And it's 10 times smaller than the standard carbon fiber electrode. As I showed you earlier with the CFE, you have this hole created in the brain tissue as well as an inflammatory response. With our microinvasive probe, we hardly detected any response. It was very hard to actually find the probes in the brain. And when we quantified the markers of inflammation around the sites that we implanted with our probe versus a non-implanted brain hemisphere, we did not see a significant difference. So then we tested these in vivo, and we were able to make measurements of dopamine with these smaller probes for over a year in the rats. And more importantly, we were able to make these measurements longitudinally, meaning the signals did not decline over time. So now I want to switch to another topic, which is combining these measurements now with electrical neural activity. And so let me briefly describe why we wanted to take these measurements of electrical and dopamine chemical activity. So we know in Parkinson's disease, there's this major loss of dopamine in the brain. We also have seen that in the clinic, there's with patients that are implanted with DBS electrodes, you can take measurements from these electrodes as well as from EEG, non-invasive sensors. And there is this huge increase, abnormal increase in beta band electrical signaling. So this is a spectrogram showing measured from a patient where when the stimulator was turned off, or for example, if their dopamine replenishing drug, they were not taking their L-DOPA drug, usually what you'll see is that there's this abnormal increase in beta band signaling, which is 13 to 35 hertz in that range. So you see a lot of red signals here. And when their stimulator is turned on, the therapeutic stimuli, or when patients are given dopamine replenishing drugs, typically you'll see most of those signals disappear. And this is also associated with an improvement in motor functioning. So this has led to a hypothesis that when dopamine goes down, the beta goes up, and the inverse. And there's also been a hypothesis that these signals are like at short time scales also inverted to each other. And so in order to probe this, we developed systems to look at these electrical signals as well as the dopamine at the same time. As I mentioned earlier, in order to measure dopamine, we use FSCV, and with FSCV, we're applying active voltages to the implanted sensor. And so you can imagine that any other electrode implanted into the brain will pick up these applied voltages because of the conductive nature of the brain tissue environment. And so this is showing a measured trace from another probe that we implanted as we were recording dopamine with FSCD. And so you can see these large spikes that occur every 100 milliseconds, which are associated with the FSCD waveform. And in order to remove this, what we did was we convert this time domain signal to the frequency domain. Here you can see the 10 hertz peak, which is associated with the dopamine sampling rate, the 10 hertz sampling rate, as well as all of the harmonics associated with this waveform. And in this frequency domain, it's very easy to interpolate away these noisy harmonic peaks. And then we can convert this back to the time domain where we've largely removed these large spikes. And then we can further remove the high frequency noise by deglitching the signal. And then we downsample the signal to about 1 kilohertz because the signals of interest are below 100 hertz. And so we've applied these in the task performing non-human primates. So this is an example of a measurement that we made as the animal was performing the task. In the color plot, you can clearly see current changes occurring at those dopamine redox potentials. And below that is the extracted dopamine concentration change traces. And you can see a lot of modulation of the dopamine signal, especially around the target and reward events, which we kind of expect based on how dopamine signals this reward value or expectation signal. We also measured the LFP at the same time, and this is a raw trace of the LFP below that. And then we filter the signal to the beta band frequency and then square and envelope the signal to retrieve the beta band power, which is typically the way that we look at beta band signaling. And we also measured pupil diameter, licking, and heart rate at the same time to better estimate the animal's internal state during this task performance. So to summarize, we developed microinvasive probes that provided longitudinally stable measurements of dopamine in vivo. We've also developed these multimodal techniques to look at the coactive functions of dopamine and beta as a function of different reward and movement conditions. The next steps is to spatially map out the dopamine functions in mood and movement control behavior. So I wasn't able to show much of this today, but we've seen that the dopamine responses can be very different depending on where we record from in the striatum. And so we want to further explore this. And we also want to look at the role of dopamine during the course of learning. And so now that we have these longitudinally stable measurements, we can now look at this day-by-day changes in learning and neurodegeneration. Thank you for your attention during this talk. And I'd like to acknowledge all the support that we received to financially fund the research, as well as all of my teammates and colleagues at MIT who were critical to the success of these the research projects. Thank you.
Helen, thank you very much for that wonderful talk. I'd like to open up the floor for questions. Please raise your hand or ask a question if you're within the panelists. All right. Helen, one of the questions that I wanted to ask you was something that you sort of alluded to in your last slide about learning. Wanted to get a sense of how you think, what type of signals you will see when you have short-term learning as well as long-term learning.
Oh, that's a great question. Yeah, that's something that I'm very interested in. So a lot of the tasks that we usually look at is the short-term learning, like reversal over a short amount of trials and how animals learn to associate different cues, right? So that's something I do want to explore. If you're asking about how the dopamine signals might be different, yeah, I can't really answer that.
Speculate, yeah, exactly, right.
There's so many hypotheses, so I don't want to say something incorrect and then and get in trouble. So. Okay,
all right. Let me ask, is there anybody else? No, okay. So let me ask one more question. You know, when we study LFPs and we do analysis on LFPs, there's always this concern about the signal that you're analyzing. Is it due to the spiking activity locally, or is it due to incoming input? And those two are often hard to separate out. And I'm wondering if, and my rudimentary knowledge is there's a lot of spiking activity in these areas as well, right? And so I was wondering if you had some thoughts on whether these bands of activities that you see, if it's more due to the incoming input or more due to the local spiking.
Yeah, that's a really great question. Yeah, there's a lot of controversy a lot of times about these LFP signals, whether they're just a result of a spiking signals, right? Integration of the spikes. So actually, when we do our processing of the signals, we do this deglitching step to remove high frequency noise, but it's also to remove the spikes directly from the recording. And so the hope is that we don't, we kind of avoid that issue. Also, a lot of our measurements that we made of LFPs are from very low impedance electrodes that cannot pick up spikes. So, so I guess an advantage of those is that we avoid that issue completely, I think. But yeah, that's a great question.
The reason I bring that up is even though the spike itself is, let's say, a millisecond in its duration, you don't know what's happening sub-threshold levels before and after the spikes, or at least after, right? And those you're probably not removing. You don't know whether you're removing those or not.
Right. Well, so the LFPs are supposed to be representing those synaptic inputs that kind of lead to that threshold. So those voltage fluctuations surrounding, what causes the actual
right, exactly.
generate in the first place. So that's part of the LFP, obviously.
Okay. Awesome. Okay, wonderful. Thank you very much. Let's get set up for the next talk. My pleasure to introduce Professor Siddhartha Sikdar. He earned his PhD in electrical engineering from University of Washington. His research interests include biomedical signal and image processing, medical imaging systems, instrumentation, ultrasound, which we're going to hear about. And he's currently the director for the Center of Adaptive Systems of Brain-Body Interactions, and he's professor of bioengineering at George Mason University. The title of his talk, Sonomyography, a new application of ultrasound in rehab engineering. Siddharth, please.
Thank you, Raj, and thank you all for allowing me to share some of our work with you. So I'm going to talk about something that's very different from what we have been hearing so far. So as Raj mentioned, my background is in medical imaging. I did my training and my postdoctoral work in ultrasound imaging, and a lot of my research is related to ultrasound imaging. So, so I'll start by briefly talking a little bit about some of the advances that are going on in ultrasound imaging right now, and then segue into how we can use that to our advantage for rehabilitation engineering. So traditionally, as all of you probably know, ultrasound is used for diagnostic imaging, primarily in a hospital setting, but increasingly it is becoming more and more used in point of care settings, in emergency rooms, in emergency disaster relief scenarios, in battlefield scenarios, and so forth. And there's also more recently been significant advances in developing these handheld ultrasound scanners, which are extremely compact. They can plug into your smartphone, and that smartphone becomes an ultrasound imaging systems. And there's an opportunity to even go smaller than that. So this has opened up a number of new use cases where we can transition away from not only thinking of ultrasound as a diagnostic imaging tool, but perhaps as the biosignal sensing tool, where you can even think about wearable ultrasound systems. So on this right, there's a example of a company that is trying to use ultrasound for looking at muscle function during dynamic tasks like running or walking and things like that. So this could become a new way of looking inside the body at muscle function during biomechanical assessments and other types of assessment where imaging has traditionally not been used. So we are trying to leverage some of these advances, and one of the use cases I'm going to talk about today is to use ultrasound for sensing muscle activity and using that for controlling external devices like prosthetic hands and exoskeletons and things like that. So the outline for the talk today is going to be, I'm going to briefly talk about the motivation for using ultrasound for prosthetic control and what some advantages are over traditional methods. And I'll share some of the results from ongoing studies. I'll talk briefly about the kind of technical advances we are pursuing in terms of low power ultrasound imaging for sort of chronic monitoring, for long-term monitoring, and really using it as a wearable sensing method. And I'll close with a future outlook for wearable ultrasound. So so the work that we have been doing for the last 5 years or so is focused on looking at the upper limb prosthetics, and this is a field with a very long history. Body-powered hooks have been used for decades, perhaps centuries, and are still in use. Many people use myoelectric control as a primary method of controlling either hooks as well as multi-articulated prosthetic hands, which are very sophisticated nowadays with multiple degrees of freedom. But myoelectric control continues to be fairly challenging. And in fact, many surveys have shown that even with these very sophisticated myoelectric hands, almost 45% or even sometimes half of the individuals who receive these sophisticated hands eventually abandon the use of this system, or they don't use it very frequently. And a lot of them state that the functionality is really not robust enough, or these systems are not functional enough for their use, even though the electromechanical design of these hands have become very sophisticated. The control has been significantly challenging. So I'll give you an example of what kind of challenges people face. So this is one of our study subject. He is a biomedical engineer himself, so he actually designed some of these very sophisticated myoelectric signaling and control strategies, which is called pattern recognition. So this is a system where there are 8 electromyography sensors inside the socket. So this is a transhumeral amputee subject. He has 8 sensors in his upper arm, and they are simultaneously recording these electrical signals. And there's a pattern recognition algorithm that is trying to discriminate between his volitional intent and using that to control this device. So you'll see that he's actually struggling quite a bit to perform this standardized task, which is called a clothespin relocation task. And some of the challenges that he's having is, first of all, he's unable to get the wrist to a particular position so that he can put the clothespin on this rod. So the ability to get the wrist to a particular position and orientation is a pretty significant challenge. And that's because electromyography uses this method of velocity control, where it's really usually an on-off type of control, where when the signal is on, you keep rotating your wrist. If it's off, then you stop rotating your wrist. And that is not very precise. It's very challenging to get to a particular position. The second is, of course, having control over the terminal device. When do you open the device? When do you close the device? That itself is pretty challenging. And so even though people have been working on myoelectric control for several decades, these challenges continue to persist. So the inferring the volitional intent of the user, not only the class of motion that they're trying to perform, but also how proportionally controlling that motion continues to be pretty challenging. So the field is sort of moving towards more invasive strategies because of these challenges. So they're moving away from the traditional mode switching, where you essentially, it's like a menu-driven method where you use these kind of EMG triggers to control the prosthetic hand, towards more implanted myoelectric sensors, neural interfaces, targeted muscle reinnervation, and so forth, and which have tremendous promise. And there's a lot of very exciting work going on in this area. But in talking to subjects, it seems like many subjects would actually prefer to continue to use non-invasive methods if there was an opportunity to do so. So our lab has been trying to see, can we come up with a better non-invasive biosignal sensing method that can do better than EMG and can overcome some of the inherent challenges with EMG. And so because my background is in ultrasound, we have been investigating ultrasound as an alternative biosignal sensing method. And so here I'm going to try to demonstrate what some of the advantages of the ultrasound are potentially over EMG. So EMG, of course, is a surface electrical signaling sensing system. So we put an electrode on the skin surface, and you're trying to infer the electrical activity of the deep muscles. So here I'm showing a subject doing two tasks. One is an index finger flexion and a middle finger flexion. And you can actually see, so this is a cross section of the forearm where we are looking at these muscle groups in the forearm, and we're trying to see, where are these activities happening. So you can see that when there is an index finger flexion, there's a portion of the flexor digitorum superficialis which gets activated. And if you're doing a middle finger flexion, a very similar area of the muscle gets activated, but actually overlying the area where the index finger flexion is happening. So if you were to use a EMG sensor at the skin surface, it'll be very challenging to differentiate between these two signals because they're coming from two muscle groups that are overlying each other. But with the use of ultrasound, we can actually clearly spatially separate these two activities because we have the depth resolution with ultrasound. So we are able to separate in depth, so that allows us to have another dimension to try to differentiate these types of signals. And of course, because with an image, you can actually get much more richer data, not only in depth, but also laterally, which allows you to spatially separate where these deep-seated muscles are getting activated. So then we can use this spatiotemporal patterns of muscle deformation and try to learn what kind of volitional movement they're associated with. So we can now use these computer pattern recognition algorithms on these image sequences to try to differentiate different methods. So, and this also works in subjects with amputation. Of course, their muscle anatomy is significantly altered because of the surgery. And so the interfaces are often, not as clearly defined, but we can still see these deformations of the muscle when they are imagining these volitional movements. So the innervation of the muscles are still present. So the muscles still deform with these volitional commands. And we can differentiate individual digits as well as different types of grasps. So, so this is a paradigm for sonomyography, which we are saying is an alternative to electromyography. So the paradigm is to put an ultrasound sensor inside the socket of the prosthetic device, and then get these kind of spatiotemporal images, and then try to use machine learning algorithms to derive control signals based on the deformation of the muscle compartments. So, and we use fairly standard supervised learning algorithms. We can do much more sophisticated things, but because we have such rich data, even fairly straightforward nearest neighbor type of algorithms seem to work well. So we train this system using a training sequence very much like myoelectric control, and then during the task performance we recognize which pattern they're trying to perform. So this is some old data that where we showed this ability to control individual digits in a virtual environment. This is an able-bodied subject. We have an ultrasound sensor, and this is a clinical ultrasound system that's looking at the forearm of the subject. And as they are moving their hand, we can decode what volitional intent they're attempting to perform. So there are a couple of things I want to point out. First is the ability to do proportional control. So you can see that they're proportionally rotating their wrist, and we can get a proportional signal. So remember that initial video I showed where the subject with the transcendental amputation was having a really hard time controlling the wrist position. With ultrasound, because we are looking at a mechanical deformation, we can actually get very good proportional control. We can also get individual signals from individual digits, which is very challenging to get an EMG. So we are now transitioning into a more compact instrumentation where we can do this with actual prosthetic device. So it's no longer just a virtual system. We can actually do this with physical devices, terminal devices as well. So in a recent study, we studied 7 subjects with different levels of amputation, with different types of amputation as well. Some were congenital limb loss subjects, others had both transradial as well as transhumeral amputation. And we, on average, got classification accuracy exceeding 85% and sometimes close to 100% in these subjects. So of course, these are offline classification accuracies for this study, but we are now showing that you can actually do task performance as well. So here's an example of a subject doing a functional task with a myoelectric direct control system. So this is a subject who has had a prosthetic for almost 50 years. So he's a very experienced prosthetic user. So you can see that as he is trying to use his terminal device with myoelectric control, he's having difficulty controlling the terminal device. And as a result, he's using a lot of compensatory upper extremity movements to try to compensate for that loss of control. This is the same subject now using our sonomyography system. So we have an ultrasound sensor in the socket, and he's using the sonomyography now to control the terminal device. And you can see that he is actually able to get more control over the terminal device. And one evidence of that he's standing up straighter. He's not using as many compensatory movements and so forth. So this is so far just anecdotal evidence. We are in the process of doing a number of careful trials of evaluating this. So those trials are right now ongoing. We'll hopefully have results to share very soon. And so this is another example of this kind of ability to do proportional control. So this is a subject who is picking up this bottle of water, and he's able to manipulate the amount of pressure he's putting on the plastic bottle to just not to spill the water, right? So it shows that we can get very fine proportional control for this kind of functional tasks, which is not something that EMG is able to do. So we have published some of this work. We have shown that we can get a very high task completion rate, as well as low sort of positional control, positional error rate for controlling a virtual cursor. So if you're trying to move as an amputee subject is trying to move a virtual cursor on a screen, we can get good positional accuracy as well as high stability for those tasks. So these are all data that have been published recently. You can see this is a real-time trace of the cursor position as the subject is moving from different levels. So now what we are trying to do is figuring out how do you put an ultrasound transducer inside a socket. So one of the observations we made is that you actually do not need this kind of high resolution ultrasound imaging system. You can actually subsample the image with just a few scan lines and get the same kind of accuracy. So we did one study where we looked at comparing the full imaging field of view versus looking at 4 randomly selected these scan lines, which we call M mode. So these are looking at depth over time. So these are motion mode images where it's one axis is depth, the other axis is time. And just by looking at these images over time, you can actually get similar classification accuracy to the fully sampled original image. So this now allows us to use very compact instrumentation where we have just 4 sensors with some very custom instrumentation that could be low power, and we could put that inside a socket, and we can get very similar classification accuracy with this compact system. So so that's where we are right now. So I'm going to conclude by saying that it appears that sonomyography is a promising alternative to electromyography, can overcome some of the challenges with EMG for signal stability, for signal to noise, and also this ability to get proportional control signals. So, and we are right now in the process of doing a number of clinical studies with several grants, as well as a number of our collaborators, clinical collaborators. So I want to acknowledge the students in the lab who have been working on this, as well as our funding sources, and I thank you for your attention.
Siddharth, that was a wonderful presentation. Thank you very much. Going to open up the floor for questions. Does anybody want to raise a hand or ask a question through the Q&A?
Looks like there's maybe one in the Q&A.
No, that's a previous conversation, if I'm seeing it correctly. Yeah.
Yep. Yeah.
Okay. I'll ask a question. You emphasized proportional control. That means you must have looked at PI control or PID control. And can you comment on, what kind of control you have with those types of controllers?
Right. So yes, so you could utilize the myoelectric signal and utilize a PID controller to try to get some kind of signal. So that's what all the commercial systems sort of do, right? So the control signal itself is almost like a on-off, whether the EMG signal is above a certain amplitude or not. And so then you get a range of velocities from that, and then the velocity is passed on to the PID controller that's in the hand. So that's what the commercial systems do. But the challenge with that is that the control signal itself is a velocity signal. Right. So so if you want to get to a particular position, like, for example, you want to rotate your wrist to 90 degrees, it is somewhat challenging to do that because your input is a velocity as opposed to a position. So because we are looking at the mechanical deformation, we can actually get a position signal. And so if the control signal is position, then you don't need to integrate up to get to that.
Yes, understood. There's a question in the Q&A from Sanjeev Shroff. It says, great technology. How sensitive is the performance to proper orientation of the transducer and proper ultrasound coupling to the object of interest? Also, what is the frequency response of the measurement system?
Yeah, so a very good question. So we are actually systematically studying this right now. Our initial results seem to suggest that it's actually not as sensitive to the orientation as we were initially worried about. And the coupling, we have some solutions for the coupling. We have been working on some materials that can essentially substitute for the traditional ultrasound coupling gel, which is not a good solution if you want to put this in a wearable system. But there are some materials that we are working on where it can be long lasting and provide the same kind of performance as ultrasound coupling gel. These are based on some thermoplastic elastomers that we we will have some data on them very soon. And you asked use 2D imaging. Yeah, so we actually don't need to use full 2D imaging, right? So we can use the single element, several single element transducers, which can give similar performance. So in terms of the frequency response, I guess you're asking about frame rate, if I'm not mistaken. Are you asking about frame rate, or are you asking about the ultrasound frequencies? The ultrasound frequencies are in the diagnostic frequency range, 3 to 10 megahertz diagnostic range. In terms of the frame rate, we can get typically about 25 to 50 frames per second. So there are some latency issues that we are trying to overcome right now, but there are ways to do that.
Thanks. Thank you very much. Yes, I was asking about the overall frequency response of your measurement, not the ultrasound frequency. So because the signal has to go down to a location, come back, and so you can only acquire data at certain see, that's what I meant, so that but your motions are not that fast, so to speak. So you capture it essentially pretty well.
That's right. You're exactly right.
Fantastic. Thank you very much. Can we have Yalda set up for the next talk? Thank you. All right, so we're going to stick with the imaging theme, and our next speaker, Dr. Yalda Shahriari. She completed her PhD in biomedical engineering from Old Dominion University, and then completed her postdoctoral training at my alma mater, UCSF. Dr. Shahriari's research interests are in biomedical signal imaging, brain-computer interface, machine learning algorithms. She's the director of Neural PC Lab, and she's assistant professor of biomedical engineering at the University of Rhode Island. Her title is a functional near-infrared spectroscopy, or FNIRS, based minimum spanning tree approach to analyze brain functional network dynamics, excuse me. Dr. Sarieri, please.
Thank you, Raj, for the introduction and organizing all of these things. So today I'm going to talk about an FNIRS-based minimal spanning tree approach to analyze brain functional network dynamics. We are going to talk especially about patients with amyotrophic lateral sclerosis or patients with ALS. So before going to the main talk, I would like to have a brief introduction of the basics. Within past several decades, brain-computer interfaces or BCIs have emerged with the initial motivation of helping patients with neuromuscular disorder, especially patients with ALS. However, later on, they showed application in other areas, including, for example, identifying biomarkers for example, for diagnosis or treatment techniques. In the closed-loop BCI, we have a feedback to the subject, and in the open-loop BCI, we basically don't have feedback. So I will talk about them in more details. The first block of all these BCI systems is a data acquisition, which can be done in two categories of invasive and non-invasive. Then the signal goes to the signal pre-processing and signal processing part, in which we clean the signal, we remove the artifacts, we filter the signal, and then we give it to the signal processing section, in which we extract the features, we select the features, and then eventually we give it to the classification algorithms that, as I mentioned, in the closed loop BCI, it can be utilized for example, controlling a device like controlling a wheelchair, doing a speller, or doing a gaming, and there is a feedback to the subject. And in the open loop BCI, or we call it passive BCI, we can utilize those classification decisions to identify markers associated with pathological conditions. So the imaging modality that we utilize in this study is functional near-infrared spectroscopy, or FNIRS. Briefly, FNIRS is a non-invasive neuroimaging modality that relies on optical technology. We place the emitters and the detector on the surface of the scalp, and it emits the near-infrared light. It passes the skull and the scalp, and it hits the cerebral cortex. And based on the activity of that cerebral cortex, that area, it absorbs part of the light, and it reflects the portion of the light. And therefore, based on this, it quantifies the oxygen hemoglobin changes in that particular area. It provides the hemodynamic changes similar to what functional magnetic resonance imaging does, or fMRI does, but fMRI has much higher spatial resolution. However, it's much more bulky and more expensive compared with the FNIRS. So this technique basically takes advantage of the optical window in which the bone, the tissue, and the skin are transparent to near-infrared light, which is between 700 to 900 nanometer. It is an excellent candidate to record from frontal and prefrontal areas important for executive functions. It works based on two wavelengths to extract oxy and deoxygenation hemoglobin level. Compared with EEG or electroencephalography, because majority of these BCI systems for ALS patients, they rely on EEG. However, these EEG-based BCIs, they suffer from low spatial resolution. So FNIRS relatively provides a higher spatial resolution. It has a higher signal-to-noise ratio, possibly because of it's less sensitive to the artifacts. And also, as I mentioned, EEG can be very challenging in recording from prefrontal and frontal area, which are very critical in executive functions. It measures another essential neural property rather than EEG. So it measures the hemodynamic vascular properties, in which EEG measures the electrical properties. And also, the good thing about it, it has the same level of portability and availability, which can be really practical for patients' bedsides, because a lot of of time patients with ALS, they cannot commute. So we have to actually take the whole system to their, for example, care centers or homes. And therefore, portability and availability is very important. And FNIRS can really provide, similar to EEG, they both can provide this portability option for patients' bedsides. So ALS is a neurodegenerative disease that affects the motor neurons in the motor cortex, brain stem, and spinal cord. So you can see in this figure that as here is a normal nerve cell and muscle, and as the patients get for example, participants get ALS, so you can see the nerves start becoming more atrophied, and also the muscles are washed out. So as the patients progress over time, they lose their voluntary muscle control, and therefore these BCI systems, especially towards later stage of the disease, they can be very helpful for these patients. Another challenge in this domain is up to 40% of the time they are being misdiagnosed. A lot of time we have had patients that they come, for example, they said they got diagnosis after 2-3 years after the onset of their symptoms. Another challenge is a lot of time we used to think this is a motor neuron disease. However, a lot of time patients develop non-motor symptoms as well, and mostly the executive dysfunctions, which can affect the BCI systems, their performance. And therefore, this system is a multi-system disease, and really little is known about the underlying non-motor neural functions. Another thing that currently it has been done a lot in terms of the connectivity in this brain connectivity in these patients. It's mostly based on the structural connectivity, or the functional connectivity are mostly based on the resting state, which basically cannot reflect the executive dysfunctions. And also the findings are a lot contradictory, possibly because of the inherent biases that these connectivity measures they can impose. So we can see a lot of contradictory outcomes for the connectivity analysis for these patients. In a recent study we published in Frontiers in Neuroscience, we investigated the functional network disruptions associated with this disease. We adopted minimum spanning tree approach, which is basically a graph-based approach that has attracted the attention of a lot of people in this domain in terms of the connectivity analysis, because there are less bias to the methodological approaches that the common connectivity approaches can impose. They are insensitive to scaling factors, and therefore any sort of, for example, transformation, it really does not affect the MSD outcome. So very briefly, MSD basically network is a subgraph that traverses across all the nodes, and it minimizes the cost or the weight links without basically forming any loop. It can project the fundamental topological properties derived from conventional methods. So if you want to formulate how we can have access to topological metrics. So very briefly, you can see here the montage. So we place the, let me activate my, okay. So you can see here we have detector, we have emitter, and the channel is the connection between these two. We record the FNIR signal, we calculate the HBO2 signal, we segment the signal, and then we calculate the phase locking value, adjacency metrics, which I will call about it, which is a measure of synchronization between two channels. And then after there, we can calculate the MSD adjacency metrics, which again, I will talk about it. And then eventually we can have access to multiple topological metrics, including degree, leaf fraction, diameter, and so on and so forth. So phase locking value, or PLV, is a measure that quantifies the synchronization level between the phases of two signals, two time series. In our case, it's two channels. It does the Hilbert transform on the signal, therefore it basically has access to the analytical form of the signal, and therefore we can have access to the real and imaginary part of the signal, and we can calculate the instantaneous phase from there. And then after calculating the instantaneous phase, phase, we can calculate the PLV by getting the magnitude of the average of phase exponential of the phase difference between two channels over the trial. So this average is basically over multiple trials. Eventually, because we are interested in the task phase, so we do a baseline normalization to cancel the phase synchronization associated with the non-task or the resting state. We utilize the Kruskal algorithm, which to form our MST, which basically sorts these PLV weights in a descending order. It starts from the highest weight, and then it continues. And if there is an edge that creates a loop, so it skips that loops, and it again do the whole thing again until it goes through all the nodes. So all the nodes are being included without creating any loop. So the metrics that we used, there were two main categories of local and global. For the local topological metrics, we looked at leaf and hops. So basically leaf is basically the node that has one edge or less than one edge, and hop is the nodes that have more than one edge. So one of the key characteristics of human brain networks is the presence of these hops, which are very important and essential for communication. Another thing is the betweenness centrality, which is another measure of centrality, and it reflects how important that node is in terms of communication with other nodes. Another measure that we looked at was eccentricity, which is another measure of centrality. However, it also demonstrates the traffic and the cost to that node as well. For the global metrics, we looked at multiple topological metrics, including the leaf fraction, maximum degree, maximum betweenness, which basically the betweenness that I explained, or the degree that I explained. So it looks at those things in the globally, so in the whole head. So globally, it takes a look at those metrics. We recruited 9 patients with ALS and 10 healthy control. They were age matched, average age of 58 for ALS and 6 years old for healthy. We adopted our previously proposed visual mental paradigm, which relies on conventional P300 paradigm, but it has a mental arithmetic task, which basically evokes executive functions as well. We recorded signal from 14 channels, primary focus on the frontal and prefrontal areas. We applied the modified Lambert law to extract the HbO2 and HbR. We applied the bandpass filter between 0.01 and 0.15 to get rid of the physiological noises caused by respiratory and cardiac activity. And then we segmented the data to the post-stimulus and the 12-second post-stimulus, 4-second pre-stimulus to do baseline normalization, and 2 seconds margins for canceling the edge effect that we later used for our phase analysis. Our group analysis showed that there is overall a higher phase synchronization for healthy control compared with the ALS patients. You can see here that the network is more evenly distributed in the healthy group than the ALS group. In the ALS group, it's more centralized towards the right, frontal, prefrontal area, so it shows more lateralized pattern compared with the control group. And you can see here also the red ones are the hops, which are, as I mentioned, the important nodes, they are more towards the right hemisphere compared with the healthy control, which is more evenly distributed across the head. Our global analysis also showed there was a significant differences between majority of our global metrics between these two groups, which also indicates more centralized global network in these patients compared with the healthy control. And also here you can see this is the hop occurrence and the edge occurrence. Again, for ALS patients, we can see the hop occurrence and edge occurrence are more towards the right prefrontal and frontal area compared with the healthy control. Our clinical association also showed that there is a significant significant positive correlation between ALS functional rating score and the global PLV, meaning that the more functional the patients are, the higher synchronization is across their whole brain. So in the other word, if they become less functional and their disease progresses, so their synchronization across their head gets suppressed. Also, there was a significant correlation between multiple of the topological metrics, including leaf fraction and tree hierarchy, which basically shows the centrality with the disease duration. So as the disease progresses, this centrality becomes more, and it was more towards right area, as I mentioned. So overall, for the first time, we demonstrated that MST analysis can mirror the frontal changes in the functional network topology in ALS during the task. So that the patients actually did some tasks, and we could evaluate some executive functions in them. So it can play a complementary role to the existing motor-related network that already a lot of studies have done. We observed a more centralized and asymmetric pattern in the brain network in these patients. So totally a more centralized network. It can be interpreted as a more random and more dysregulated network. And also the information traverses along fewer and more overload nodes, which again can be the reason to have, for example, disruption in the quality of information communication in these patients. And another interpretation can be related to that we had more density to the right prefrontal area, because they want to do some sort of compensatory attempt to control over the task, and therefore those parts in the right area gets more activated. And this actually hyper-excitability mechanism in the right area, it can also be interpreted to compensate for the structural atrophies supported by a lot of structural ALS studies. At the end, I would like to thank my students in my lab and the funding supported our study. Thank you.
Thank you, Yalda. Time is tight. Does anybody have a quick question? Okay, I'll ask a very quick question here because poster session's upon us. Oh, did somebody just? Okay, can you comment on variability? So all of these network connectivity disorders, there's a lot of variability from subject to subject, from patient to patient, excuse me. Can you comment on how robust the analysis is to detecting those kinds of variability?
Yes, so that's the good thing about MST approach, because it's not really sensitive to the amount of the weight. So it basically, as I mentioned, it is sensitive to the order. So any sort of transformation, any sort of changes in those weights, it really does not affect. And this MST approach is really perfect if you want to compare like different cohorts or different, for example, conditions. So this is like much less biased compared with other connectivity measures.
I see. All right. Thank you. All right. I hear somebody speaking. Is somebody wanting to say something? Oh, okay. Sanjeev, I have to unmute you.
I guess we are running late. So a quick question. If I understand the technology correctly, the signal comes from or is determined by two processes. One, the blood flow itself and the neural activity associated oxygen consumption. If that is correct, if my understanding is correct, then my question was, what if the contaminating effects of blood flow aberration itself? How does that matter? Does it matter to you at all?
So basically any sort of neural activity, it can have electrical changes, magnetic changes, and the vascular changes. So this technology basically it measures the hemodynamic vascular change in the blood oxygenation level. So as we do the activity in, for example, some sort of activity and some part of the cerebral cortex get activated. So this technique measures that activation based on the emitted, there is that Beer-Lambert law that I explained, so I didn't put show the formula, these things here, but it measures the proportion of light that has been detected to the light that has been emitted. So based on the activity in the cerebral cortex, whatever, if it is more activated, the consumption of the oxygenation level, it gets more. So then therefore the oxygenation hemoglobin levels start decreasing. However, it's at the very beginning, but then it starts coming back. I don't know if I addressed your question.
I think, yeah, maybe we can talk later. Yeah.
Sure.
Okay, thanks.
All right. Thank you very much to the speakers of this session. Appreciate very much for you taking the time and teaching us a bit about your work. Within less than 10 minutes, the second parallel research session will start, and we hope to see you there. Thank you very much.